Hello and welcome back to Simply Biology. Today we're going to be focusing on nucleic acids or nucleotides. In today's video we're going to know what a nucleotide is, the components of a nucleotide, how nucleotides are joined together to form polymer molecules, the different bases that consist of nucleotides and whether they're purines or pyrimidines. And without further ado, let's get started. So, a nucleotide is a monomer which can be formed and joined together to form a polynucleotide, which is the polymer. So DNA is two polynucleotide strands. So polynucleotide is the polymer that is formed by many repeating monomers, which are when the monomers are the nucleotides. So the nucleotide is composed of three separate components. You've got a pentose sugar, which can either be ribose in RNA or deoxyribose in DNA. The phosphate group, which is slightly acidic and negatively charged part of the nucleotide molecule. And then you've also got a nitrogenous base, and obviously these bases are complementary, with adenine pairs with thymine, guanine pairs with cytosine in DNA, and then you've also got uracil, which pairs with adenine in RNA. So these are the three components of the nucleotide, and there's slightly different components for either DNA or RNA. So we're now going to look at the components of the nitrogenous base and whether they're purines or pyrimidine. So purines consist of adenine and guanine and have a double aromatic ring structure. This means they contain two rings in their structure, in their chemical displayed formula, whereas pyrimidines consist of thymine, uracil and cytosine and only have a single ring structure. We're now going to look at how you can join nucleotides together. So nucleotides are joined together by condensation reactions, which include the elimination of water. So here's a very simplified structure of a nucleotide. So this is the phosphate group. This is the ribose sugar, which can either be deoxyribose or ribose. And then you've got the nitrogenous base, which can either be A, C, T or G, and U if it's an RNA molecule. So here we have the displayed formula of what's known as the nucleotide. So this is just the chemical displayed formula of the nucleotide, and this here is the nitrogenous base for thymine. So as you can see, thymine is a pyrimidine and it only has one aromatic structure. So this is what I was referring to in the last slide. So here you can see the hydroxyl group of the sugar and the H from the thymine base are added and they're removed in order to form this bond here through a condensation reaction. And this same thing also happens between the phosphate group here and the ribose sugar here. So the hydroxyl group is taken from the sugar and the H is taken from the phosphate group, and again another molecule of water is removed in the process of the condensation reaction. And this overall molecule is what's known as the nucleotide. So finally, we're going to look at ATP. So ATP is a special nucleotide which consists of adenine, a ribose sugar, and three phosphate groups. Now, it's known as adenosine triphosphate or ATP, and it is a universal energy carrier and a short-term supply of energy. So adenine is the nucleotide and it's phosphorylated, meaning it has three phosphate groups attached. So ATP is an energy source and it releases energy when it's hydrolyzed to form ADP or adenine diphosphate, and it can be either further hydrolyzed to form AMP, adenine monophosphate. So just two phosphates or monophosphate signifies one phosphate group attached to adenine. So what can happen is the ADP or the AMP can be restored to ATP during a process of phosphorylation. So, and as just said, ATP consists of a ribose sugar, an adenine base, and 3 phosphate groups. So it's also important to note that all the bases are nitrogenous bases, meaning they contain nitrogen in their chemical formula. Well, thank you for watching this video, and I hope you've learned something new about nucleic acids and their role as monomers in the production of larger polynucleotides molecules. Thank you. Goodbye.